Hello, good morning everyone. My name is Sagnik Mitra. I am a PhD student at Department of Biosciences and Biomedical Engineering at IIT Indore. So today I'll be talking about genome scale metabolic reconstruction. So genome scale metabolic reconstruction is an in silico tool or an application in which different organisms are actually like the metabolic construct of each of the organisms are modeled in silico, and they are used for like industrial applications for in terms of like product formation or product development, etc. So in this genome scale metabolic reconstruction, what we basically do is uh, we construct a model for an organism, and we try to understand the metabolic pathways associated with that particular organism, and we try to tweak into those metabolic pathways in order to understand what kind of products that could be beneficial to us and could be used for industrial applications as well. So in this slide, I have mentioned about like if we do a metabolic reconstruction for a particular species or an organism, we'll be able to estimate the following, which is to predict the culture conditions and possible responses to environmental changes, predict metabolic capabilities from genotypes, and predict the impact of genetic perturbations. So we will look into details of these conditions in the subsequent slides. So in this topic, I'll be covering constraint-based modeling, flux distribution, constraints on biological systems, stoichiometric matrix, flux balance analysis, choice of objective functions, and perturbations to metabolic networks. So all these topics will almost summarize the fact that what kind of mechanisms are involved in the genome scale metabolic reconstruction. So first I will look into the constraint based modeling. So constraint based modeling is basically like suppose we have a model for an organism, like model in the sense like model will comprises of basically, like if we go mathematically, it is basically a range of matrices that is formed, like it has rows and columns with different reactions and metabolites and different reaction names and everything. And from those matrices we will be able to subject that particular model to some certain constraints. So that constraint could be in the form of, like, suppose we restrict the particular metabolite and see how the pathways are getting used up in the whole metabolic network. Suppose we want to restrict the growth of the organism and see how the pathways are getting regulated. So this is one of the constraints we are subject. So basically, with the help of those constraints, we will be able to understand what kind of things are happening inside the particular organism and how our product of interest is getting formed. So any constraints govern possible biological functions. That is true. So any kind of constraints that you give into those organisms. So in any kind of systems biology or biological equipments or biological organisms, you'll be able to find that if you it is basically easier to understand any biological system by doing some either removal of things, like suppose you remove a genetic circuit or remove a biochemical component. So these kind of things, so each of these things are kind of constraints in certain manners. So any constraints govern possible biological function. Organisms exist in resource scarce environment. So if you limit the amount of glucose for a certain organism, so that is a resource scarce environment for that organism. More fit organisms survive with higher probability. Fitness requires satisfying several constraints, limits range of phenotypes, and there are models. Okay, so one thing I would like to mention is that there are different types of modeling. So this is one of need. So this constraint based modeling comes under the domain of kinetic modeling. So there is alternatives to kinetic modeling I have mentioned here. So survival of the organism depends on the best utilization of resources to survive and grow and subject to constraints. All phenotypes must satisfy constraints. Constraints can help eliminate impossible behaviors as well. So this is the basic understanding of constraint based modeling. Flux distribution. Flux, so in so metabolic pathways of any organism, a flux is typically the amount of material that is flowing from one place to another. So flux is basically an amount. It represents an amount in which suppose if we go into the glucose pathway, glucose or the glycolysis pathway, the amount of glucose that is getting consumed by each of the organisms, different organisms will have different amounts of taking up the glucose or utilizing or uptake of the glucose. Okay, so that will help us understand what kind of organisms we are dealing with, what kind of sugars they can take into, which could be in the form of glucose, which could be in the form of fructose, which of the sugar molecules are they best utilizing. So I'm giving the example of glucose here. It could be fructose, it could be galactose. So all these are 6 carbon sugars. Some organisms are suited to 5 carbon sugars as well. So those kind of things so how much amount they are taking up, that is the flux distribution. So in this context, flux distribution refers to the amount of metabolites in all the metabolic pathways and how they are distributed over the entire network. So the rate of change of flux distribution can be represented as follows. So suppose there is a reaction 1 which represents A plus B converts to C, and there is reaction 2 where B plus C converts to D, and there is reaction 3 which represents C to E. So the first reaction's rate is V1, second reaction rate is V2, and the third reaction rate is V3. So all these V1, V2 and V3 are the flux distributions over the entire reaction network. Okay, so constraints on biological systems. So there are different kinds of constraints which could be in the form of environmental constraints, could be pH, temperature, osmolarity, availability of electrons, receptors, availability of carbon, oxygen, sulfur, nitrogen, phosphate sources. There could be regulatory constraints which could be subject to evolutionary change, which allows the cells to eliminate suboptimal phenotypes and confine themselves to behaviors of increased fitness. There could be physicochemical and system-specific constraints as well, which could be in the form of connectivity, thermodynamics, capacity, kinetics, and genetic regulation. So the basic understanding, mathematically, the basic understanding of flux balance analysis is the understanding of the stoichiometric matrix. So stoichiometric matrix is basically the reactants and the products in a reaction can be represented in the form of a matrix format, which makes it easier to solve as well as to understand. So the stoichiometric matrix S is represented as S M cross N, where M is the metabolites and N is the reactions. So the metabolite concentration is represented as X, whereas the V represents the reaction fluxes. Therefore, the rate of change of metabolite concentration can be represented as dx by dt equals to S into V, which represents the equation 1. And for the steady state mass balance, there should be no rate of change of the fluxes or the concentration. So dx by dt equals to S into V equals to 0. So this equation gives rise to a system of linear equations which could be underdetermined, overdetermined, and the exact system. So undetermined means M should be less than N. Overdetermined means N should be greater than M, and the exact system is M equals to N, where metabolites and reactions should be equal. That is the exact system. So FBA typically, so in another way, FBA is typically refers to solving a constraint-based optimization algorithm to understand the movement of metabolites in certain pathways of interest and how it is affecting the organism at phenotypic level. Another way to quantitatively define FBA is solving a linear programming problem that would get us the values of what kind of flux distribution is taking place in the whole reaction network or the metabolic network. So to get the FBA solution, we need to understand what kind of objective functions we are going to apply in order to get those particular values. So that depends on the desired goal of the simulation. So that helps us in basic exploration and probing of solution space, to represent likely physiological objectives, and to represent the bioengineering design objectives. So choice of objective function is very important in order to understand the kind of FBA solution you want for the organism or the model, to be precise. Now, perturbations to the metabolic networks. Okay, so a fundamental philosophy, as I was talking before, of systems biology is to study and understand the biological systems through perturbations. Metabolic networks can be perturbed primarily by the removal of genes or the overexpression of genes. The effect of such changes in genotypes can be simulated using constant-based analysis techniques such as FPA, MoMA, and ROOM. So FPA, MoMA, and ROOM. So MoMA and ROOM are another two different forms of constraint-based analysis. For metabolic engineering, we are primarily focused on identifying genotypes that have improved properties. For drug target identification, we are interested in identifying proteins which when removed, have a serious deleterious effect on growth or survival of the pathogens that we are taking in as model. So conclusion, constructing such genome scale models, especially for any organism for industrial production of products, is of utmost importance to any industry as it will save lots of money. Performing API studies will help us understand the flux distributions and will help us in enriching the updating and training the model based on the kinds of constraints used. GSMM will pave the way for understanding eukaryotic organisms as well, like fungi, which is gaining much popularity, and also developing updated models of existing organisms is still helpful, which can be explored for experimental verification by MFA studies. Thank you.